Alrighty guys, so today we're just kind of going through a quick recap of what we've been doing the past couple weeks just to make sure we're all up to speed. And as you're going through your study guide, you can kind of like at least get some of this stuff knocked out, and then again you can always go back to your other videos as well, whatever helps. So let's start with at the beginning with meiosis. So meiosis is going to be a process. Whoops, I didn't mean to make a text box. I'll just delete that over there. Go away. All right, so meiosis is going to be the process of creating something that we call gametes. At the end of the day, what gametes are is they're going to be sperm or eggs that are going to eventually fuse to form a new individual. So basically, you get half of the genetic information so that your organism can basically reproduce in a way that you are mixing up the genes, okay? So now we're moving on from something where you're just creating copies. Now we're getting genetically unique individuals. That is the point, that is the overall goal of meiosis. So the overall goal, again, is you're gonna get genetically unique individuals at the end of the day by getting this cell where you have half of that information, and then later you can fuse those cells and get something completely different. So how does meiosis happen? So some important things to keep in mind is meiosis is not going to happen in every cell. They are only going to happen in what we would call the germline, or well, I'll just put germline for now. But basically your reproductive cells. Skin cells cannot do this. Also keep in mind your bacteria friends cannot do this either. It's only going to be in eukaryotic cells that are going to eventually form gametes. So the process of meiosis is it's going to have two overall separations. Basically, if you remember all the way back into mitosis, we called that PMAT, right? So it was prophase, metaphase, anaphase, and telophase. Basically, you're going to do PMAT twice in meiosis because you want to get half of that genetic information. So here, the difference here in meiosis 1, okay, is where some unusual stuff is going to happen. That's what we call the first division. In meiosis 1, we can have a phenomenon that happens called crossing over, okay? So crossing over is when part of those DNA segments, if you see here, part of those DNA segments are flipped, and they basically, when the homologous chromosomes get really, really close together, they can actually switch some of that genetic information. So what the other key thing that you're going to be able to tell the difference between meiosis 1 and meiosis 2 is going to be what is happening at anaphase. So at anaphase in meiosis 1, so I'm going to put anaphase, you have homologous chromosomes separating, okay? That is the main idea here, is you have homologous chromosomes separating instead of what it looked like in mitosis where they kind of kept together. In meiosis 1, the way that anaphase kind of looks is you have the X's kind of in two lines like that. So that's going to be a key difference there between meiosis 1 and meiosis 2. Then in meiosis 2, it basically looks the same as what it did like in mitosis. So one key thing that I'm going to write over here, I'm going to erase this over here, is meiosis 2, okay? Meiosis 2, you are going to, in anaphase, instead of separating homologous chromosomes, you are separating sister chromatids, okay? That is the main goal there. So at the end of the day, you end up with 4 cells. They got kind of cut off on this image, but you're going to get 4 cells. Each of those 4 cells is going to have half of the genetic information. So now they are ready to go. You could theoretically use those to form a new individual. All right, so the next thing that I want to make sure that we go over is going to be DNA structure. So some key things to keep in mind with DNA structure is that it is shaped like a double helix. So it's shaped like a double helix, which it kind of looks like a twisty ladder. It looks like this. And the building blocks are called nucleotides. Now, nucleotides are made up of 3 parts. You have your phosphate, you have a sugar, and you have a base. So that base is going to be what really matters in terms of how we store our genetic information. Because the base, we have 4 types of bases. We have adenine, we have thymine, we have cytosine, and we have guanine. Now there's a special rule that we use to figure out what goes with what, and that is that A is always going to match with T and C always matches with G. So those are the base pair rules that help us figure out how we would match this strand together. All of these bases are going to be right in the middle of the DNA, where kind of the rungs of the ladder is how you would see it. And then the phosphate and the sugar are going to be the backbone. So they're going to be the sides of the twisty ladder, so to speak. So that is the basic overall structure. You can look more into that on the past videos as well. All right. So next we have our DNA processes. So there are 3 main processes that I want to make sure I highlight today. And I'm going to kind of leave the review here. Even though we added in mutations, I'm just going to kind of leave it here because you just watched the video on mutations, so it's not that old yet, but I wanted to make a quick recap here. So replication is going to be making more DNA. The idea here is that you need more DNA, and you need this before you do mitosis, you need this before you do meiosis, all of that stuff you're going to need to make a copy. Transcription is going to be going from DNA to RNA. Particularly, you have 3 types of RNA, and you're going to, in this case, be talking about mRNA. Now, keep in mind, you will need to know some differences between RNA and DNA. I'm going to put that over here on the side. So DNA versus RNA. So keep in mind, the bases are going to be different. So in DNA, you have thymine, and then in RNA, you have uracil. Double versus single-stranded is going to be something that you could talk about. And the final thing that you could talk about that is a difference is going to be the location you find these in. RNA is going to be found outside of the nucleus, whereas DNA is primarily going to be inside the nucleus. Finally, we have our last process, and that is translation. So translation is taking that RNA message and it is making a protein out of it. So you're going to make a protein at the end of the day. That is the goal. So the central dogma of genetics, the central rule, again, I think I've put this in every single video for the last couple. So just make sure you know this. DNA codes for RNA, which codes for proteins. Well, I can't spell. Okay. Either way, but DNA codes for RNA, which codes for proteins. That is the main overall goal. Transcription is going to happen in the nucleus, and translation is going to happen in the cytoplasm or the ribosomes, you could say, but it's going to happen really outside of the nucleus, you could say. Replication is also going to happen inside the nucleus. So that is a quick and easy just review session. I want to make sure that you guys have just something where you can just kind of get a general feel for some of the main outlying things. Again, this was meant to be a quick video because I wanted you to look at the practice test and all of that and make sure start going through your study guide. All right, guys. Let me know if you have any questions. I'm just an email away, and I can Zoom, I can do whatever. Just let me know. Yeah, have a good day.